As I was presenting, so in the following presentation, we will see together another application of metagenomic analysis as new approach for monitoring aquatic microbiome in Africa. So we will cover together different points. So we will see water resource in Africa, which is about quality, climate change impact, then quality and public health. I will introduce the metagenomic approach in few lines, and then we finish with some recommendation for a quality monitoring and restoration. So as we know all, Africa is home to about 30% of the world population, and it's considered as the most dry continent in the world after Australia. So already 14 African countries are facing water stress. The freshwater resources in the continent are divided into 17 big rivers and 160 lakes, which are used especially for agriculture and domestic activities. So altogether, they are impacting water qualities in most areas of the continent, which led to a serious public health issue. Almost half of the population suffers from one of the 6 major water-related diseases, such as cholera. The microbial communities in water resources, such as bacteria and archaea, are playing a key role in biochemical cycles. So these communities are able to modify their metabolic spectra in relation to different environmental stress. Based on this, they have been used as bioindicator of environmental change. However, only 2% of these bacteria can be studied using classical culture. But recent advance in molecular technologies has opened new possibilities to better study these parts of uncultured bacteria using bioinformatic analysis. Also, we can use statistical tools to perform correlation between these bacteria and environmental parameters, which can help us access the effects of water pollution on microbial composition and distribution. So few studies have been done on the African continent using this metagenomic approach. So here, using metagenomic approach. So this paper or this study have been done on water samples collected from an extreme environment in Portugal. So it's a case study that we have done together. So samples of water have been collected from this site using 16S approach. So we had possibility to study the prokaryotic communities of this water body. The results of this study indicate how this technique could be useful to access the whole microbial diversity, and how we can use specific taxa as indicator of health status. So if you need more information about this paper, you can text me after the presentation and I can answer all your questions. Thank you so much. It's all.
Thank you very, very much, Sara. So I have a question that I would like to ask you. And so you guys are using metagenomic approaches to understand the microbial taxa within these water environments. Do you think that the next sort of step to understand the functionality within these environments is to do metatranscriptomics and perhaps metaproteomics?
Yes, sure, but not using the 16S approach. So we need to do like the whole metagenomic sequencing the whole genome. You understand, not only the 16S or the 18S. And based on that, we can perform metabolic analysis, proteomic analysis, but we need the whole genome, not only the 16S. So in this presentation, I was presenting only the 16S part, but we can also do or sequence the whole metagenome, not only the 16S.
Okay, thanks, Sara. So I don't see any questions in the Q&A, which gives me an opportunity to ask another one. I just wondered if it would be worthwhile to have comparative data. So say, for example, if you were going to do the whole genome analysis, or even at the 16S level, do you think it would be very useful to compare environmental data to what you're seeing, especially if you're seeing changes over a period of time in those ecosystems?
Absolutely, yes. Yes. So if we do like a correlation between environmental data, which could be pH, concentration of some physical and chemical parameters in relation with the environment, in relation with the microbial diversity, yes, absolutely, we can get some information about how this microbial diversity could be changed or impacted by the environmental change or the physicochemical parameters of the environment of the ecosystems.
Okay, great, because I think that is sort of a very good way to approach a more holistic view of what is going on than rather just looking at who's there. And also it would be nice to see who's there over time and what kind of environmental factors are contributing to that taxonomic diversity. One more question that I have for you, and it's a bit of a sort of sideline one, is, sorry, I'm completely hogging Q&A here. So the other question that I have for you is, when it comes to sampling, what sort of measures does one need to put in place to ensure that your samples remain of a certain quality? So if you're doing analysis, or if you're doing whole genome sequencing, or if you're looking at those samples later on for different research questions, what sort of things should we put in place to make sure that we're not affecting the results via our sample storage methodology?
So whatever, if we are looking on samples of water, fresh water, for example, or even on sediments, because I did another part of the Portuguese papers have been done on sediment samples. So even that or the other, we have to respect some conditions to not impact the results by the end of the bioinformatic analysis. So during the sampling, the transportation of the samples to the lab, even during the DNA extraction. So we have some different points or some different protocols that we need to respect in order to not impacting the result by the end.